Assalamu alaikum and welcome you all to this YouTube channel, The Science Magazine. Today's topic is part of the lecture series on class 12th botany. We have already started unit 2nd of class 12th botany, that is reproduction in angiospermic plants. In previous lecture we introduced what is reproduction, what are its types, and finally we discussed asexual mode of reproduction. In today's lecture we will discuss sexual mode of reproduction in angiospermic plants. Sexual reproduction. Sexual mode of reproduction in angiospermic plants. What is sexual reproduction? Sexual reproduction is the phenomena of producing the gametes, and those gametes later on fuse with each other, resulting in formation of zygote. That means we can say that a sexual mode of reproduction is that mode of reproduction in which parentus, that is sporophytic body, reproduces haploid cells, haploid gamete cells by means of meiosis, and those haploid cells of opposite sex later on fuse together, resulting in formation of zygote, which finally develops into a new individual. Right? Sexual reproduction involves sporophytic cells gets converted into gametophytic cells. Sporophytic cells are usually diploid in nature. That means diploid cells gets converted into haploid cells. How diploid cell gets converted into haploid cell? By the means of meiosis. That means it involves the process of meiosis. That means it is reduction division. Diploid organ cells get converted into haploid cells. Those haploid cells are opposite nature. That means opposite sexes. Male haploid cell and female haploid cell fuse together, resulting in formation of diploid cell. That is, it is haploid, this is haploid, these male and female haploid cells fuse together, resulting in formation of diploid cell, that is known as zygote. And this fusion of male gamete and female gamete is known as fertilization, right? This is known as fertilization. That means sexual reproduction involves formation of gametes. Sexual reproduction involves the process of meiosis. Sexual reproduction involves the fusion of male and female gamete. Sexual reproduction involves the formation of zygote. In contrast, asexual reproduction was not involving these processes. We have seen in asexual reproduction, part of the parent plant directly gets converted into new individual. But in case of sexual reproduction, parent plant first produces gametes, that means haploid cells. Those haploid cells of opposite sexes fuse together, resulting in formation of zygote, which later on result in formation of new individual. Now you got an idea what is sexual reproduction. We are talking about angiospermic plants. In angiospermic plants, they have a special structure for the act of sexual reproduction. That organ which is involved in sexual reproduction in angiosperms is known as flower. What is flower? Flower is actually a dwarf modified shoot. Normally this is a plant bearing nodes. From nodes usually arises branches from the axil of small leaf, that is known as bract. When plant gets mature, instead of developing branch, it develops a dwarf branch. Means short branch, reduced branch. They get condensed at its terminal position. Internodal distance get reduced and a bulging like structure appears at its tip. This bulging is known as receptacle. Or thalamus. Right? From this thalamus arises different whorls of leaves, which form at its early stage, forms a bud-like structure. We call this bud-like structure as floral bud. When this floral bud gets matured, it opens into a structure like this. This flower is normally of 4 parts. Outer part, which is known as calyx, usually green in color, and individual unit of this calyx is known as sepal. This calyx protects the floral bud at its early stages from any physical damage. Second is colored part, that is known as corolla. Right? It is colored part, and individual unit of this corolla is known as petal. This corolla is colored. For what purpose? The corolla is colored because it helps in attracting pollinators for the act of pollination. These two whorls have no direct role in reproduction, sexual reproduction. That means they are not involved in the act of sexual reproduction. Calyx and corolla have supplementary role. That means calyx protects at bud stage, while as corolla attracts while opening the insects or pollinators for the act of pollination. But they are not involved directly for the development of male and female gamete or their fusion. That is why we call them as non-essential whorls. These are non-essential whorls. Non-essential whorls or accessory whorls or sometimes supplementary whorls. After that, we will talk about a male gamete producing organ which is known as androecium. This is like this, hammer-headed and a stalk-like structure. This is filament, and terminal portion is bulged out into a bilobed structure which is known as anther. It is usually bilobed, and within each lobe there are 2 sacs which are known as microsporangia. Right? That means in total 4 sacs are present within single anther. That means it is tetrasporangiate. If we dissect this anther, we can find it like right. Within these sacs are pollen grains, which are known as pollen grains. That means male organ consists of 2 parts. One is filament, second is hammer-headed or bulged terminal portion. That terminal portion is known as anther. Within anther there are 2 lobes. This is one lobe and this is second lobe. That is why we call it as bilobed. But within each lobe there are 2 sacs. We call it as pollen sacs or microsporangia, right? In total, there are 1, 2, 3, and 4 sacs in 2 lobes. We call it as tetrasporangiate condition, right? These pollens are male spores. Pollen grains are called spores. These are haploid cells. These haploid cells later on further divide, resulting in formation of 2-celled stage. One is generative cell and second is vegetative cell. This generative cell later on divides and results in formation of male gametes. Now finally the female part, that means egg producing organ, that is known as gynoecium. Male producing organ is known as androecium, which consists of 2 parts, filament and anther, whereas female producing organ is known as gynoecium, which consists of 3 parts, which consists of a receptive part which receives pollens, connecting part, and a bulged part. Upper receptive part which receives pollens from androecium, this part is known as stigmatic surface or stigma. It is known as stigma, right? Basal part which is bulged is known as ovary. Right? And the connecting between stigmat and ovary is a cylindrical or funnel shaped structure which is known as style. This is known as style. Right? That means gynoecium female gamete bearing organ consists of 3 parts. One is ovary, that is basal bulged part, and second is style, which connects this basal bulged part to the stigmatic surface, which is a receptive surface for pollens. Right? Within this ovary there are structures, small structures. These structures are known as ovules. And within these ovules there is an egg apparatus or egg cell. In some species there is a single ovule, but in some species there are more than 2 or 3 or even hundreds of ovules. Now this Individual unit of androecium is known as stamen. Same way, single gynecium is known as carpel. Right? This is overall structure of flower. It consists of 4 parts. Outer 2 are calyx and corolla, which are non-essential parts. They have no direct role in reproduction. While as those organs which have direct role in the act of sexual reproduction are androecium and gynecium. Androecium is male gamete bearing organ and gynecium is female gamete bearing organ. Androecium consists of 2 parts. One is filament and anther, while as gynecium consists of 3 parts. One is ovary, style and carpel. Right? Now, development of pollen sac or male gametophyte, that is development of microsporangia. Microsporangia are the parts of microsporophyll, that is androecium. This is stalk part and this lobed bulged terminal part is known as anther. When we cut this anther and cut it into transverse sections, we will find these are the layers of cells developed around this sac. And this is known as connective. We cut this anther into transverse section and visualized it under microscope. We find this tetrasporangiate structure, that means structure which bearing 4 sacs. These are sacs which are known as pollen sacs. Within these pollen sacs are present grains which are known as pollen grains. These are pollen grains. If we magnify this portion of this anther, it will appear like this, right? Outer single layer, which is known as epidermis. Inner to epidermis, there is a layer of cell which is known as endothecium. Endothecium. These are cells which have fibrous thickenings. They are thick, but at the position of dehiscence, through which pollen grains come out, at that position, these endothecial cells are not thickened. They are thin-walled cells. That position is known as stomium, right? That's known as stomium. Inner to this endothecium, there are few layers which are known as middle layers, right? And finally, there is a single interior layer which is known as tapetum or tapetal layer. And inner to the tapetal layer, there is a sac in which there are pollen grains. This is actual mature structure of this anther, right? How this anther gets developed to this mature stage, that means that is known as by development of pollen sac or development of anther, right? This is normal structure. This is stalk. On this stalk, there is a mass of cells covered by an outer layer of cell. These mass of cells are dividing or meristematic cells. This outer layer is known as epidermis. From this initial stage, small divisible meristematic cells get modified into a well-developed structure that is known as development of pollen sac. Now we will discuss how such a mature structure gets developed. First, at the initial stage, this whorl, that is microsporophyll or androecium, at its terminal position develop a mass of cells. Those cells are meristematic in nature, that means dividing cells. These dividing cells develop outside a layer which is known as epidermal layer. These mass of cells bulge out into a structure which appears to be two-lobed structure. Some cells get differentiated or distinguishable from rest of the meristematic cells by developing dense cytoplasm and side by side having conspicuous nucleus. These cells which develop dense cytoplasm and having conspicuous nucleus are destined to form the whole structure of anther. And those cells are called archisporial initials. Those cells are called archisporial initials. Now, if we look at this part, this is like this. This is outer layer, which is known as epidermal layer. Within the epidermal layer, there are mass of cells, but one cell gets dense cytoplasm and evident nucleus, that is known as archosporial cell. This archosporial cell first divides periclinally. That means it divides parallel to the wall, epidermal wall. This is epidermal wall. If the cell divides parallel to this, that means it forms two cells. It forms, one is towards the wall and another is away from wall. Like this, one towards the wall and second away from the wall, right? Two cells. This cell gets divided into two cells. This is known as parietal cell, and this is known as sporogenous cell. This parietal cell is responsible for the production of all the layers except epidermis is already formed. Other layers, that is endothecium, middle layers and tactile layers, develop from parietal cell, while as sporogenous cell results in formation of pollen grains. Development of different layers from parietal cell is known as development of pollen sac, while as development of pollen grains from sporogenous cell is known as microsporogenesis. Right? Here we will start with parietal cell. How parietal cell divides and results in formation of all the layers, ranging from endothecium, middle layer to tactile layer, right? This is now epidermal layer already formed. Under this layer, hypodermal cell, that is parietal cell. In reality, they divide simultaneously. Make it easy to understand, we will first start with parietal cell and later on we will discuss sporogenous cell. This parietal cell divides simultaneously, periclinally as well as anticlinally. That means it divides parallel to the wall. Anticlinally means perpendicular to the wall. One is parallel to the wall, second is perpendicular to the wall, right? Periclinal divisions are followed by many anticlinal divisions. This is parietal cell. This first divides periclinally, results in formation of two cells. These cells later on divide anticlinally. From 4 cells, there results in formation of 8 cells, forms 16 cells. They further undergoes periclinal division. This one and this one results in formation of 4 layers, and simultaneously divide anticlinally, results in formation of layers. Continuously periclinal as well as anticlinal. Outer layer, subepidermal in position, the layer formed subepidermal in position is known as endothecium. The cells of this endothecium are having fibrous thickenings, right? They have fibrous thickenings on their walls, except at the position of dehiscence, through which these pollens come out. At that position, the cells of this endothecium are thin walled. Inner to this, there are few layers which are known as middle layers. And finally, there is a layer that is known as tactile layer or tapetum. Tactile layer is nutrient function. Sporogenous cell also divides results in formation of mass of